Good afternoon, everyone. It's a pleasure to me to introduce today's speaker, Dr. Inbal Mor, Principal Investigator at the Department of Molecular Biology at Ariel University in Israel. Dr. Inbal Mor earned her PhD in biochemistry from the Ariel University of Jerusalem and went on to complete her postdoctoral training in Cross-Care Boston Lab at the Cancer Research UK Beeston Institute in Glasgow, focusing on cancer metabolism. In 2015, she returned to Israel, shifting her research towards liver diseases, particularly to the metabolic mechanisms driving muscle D. Today, at Ariel University, Dr. Mor leads a lab dedicated to studying how metabolic alterations contribute to muscle D progression and affect drug metabolism in the liver, with the goal of identifying new therapeutic strategies. Please join me in welcoming Dr. Inbal Mor. So the audience is yours.
Thank you. Thank you for the introduction, and thank you, of course, for the invitation. I'm very happy to share our research with people interested in muscle D. I'll share just a second.
Yeah.
Share my screen.
I will close my.
Oops. Well, now it's not after we tried everything before. Can you see my screen? Yes. Great.
Yes.
Yes.
Perfect.
Perfect. Okay. Good. Okay. So I'll just dive in. So I think this audience here needs no convincing that mast D is an important research field. It affects a third of the global population, with the incidence continuously rising since it was first identified decades ago. And it's therefore become the leading cause of end-stage liver disease, huge economic burden, health burden, and it's also due to become the most frequent indication for liver transplantation in just a few years. So definitely a worthy topic of study, and if I may also add, a very interesting topic to study. So as we all know, this is a progressive disease. It starts out from simple steatosis, which means there's lipid accumulation in the tissue in hepatocytes, and it can then, this chronic accumulation of lipids can then lead to more severe tissue consequences with cell damage leading to inflammation, tissue damage, fibrosis, and this can then progress and affect liver function, ending in cirrhosis, which can be a risk factor even for another end stage, which is a hepatocellular carcinoma. The same as the steatohepatitis itself is also a risk factor for liver cancer. I would actually like to focus on the earlier stages of the disease, which are reversible, definitely simple steatosis, and also early stages of hepatitis, of steatohepatitis. This stage of simple steatosis, which is very common in the population, is asymptomatic and therefore grossly underdiagnosed and truthfully also considered to be clinically benign by the medical population. So it doesn't get much attention. But if we think about this process of mast cell developing and progressing from a metabolic and a biochemical point of view, which is the way I look at the world, This characteristic phenotype of early stage disease with the lipid droplets must mean that there are considerable metabolic changes in the tissue, in the cells, even if this is clinically benign and does not affect clinical parameters of liver function. We obviously know that fatty liver disease does what it says on the tin. The liver does become fatty, which means from a metabolic, bioenergetic point of view, that the tissue is in a positive energy balance. There's more lipids coming in, either from the diet or synthesized or from adipose tissue, or synthesized de novo in hepatocytes from carbohydrates and sugar, more than the liver cells are able to eliminate, either through oxidation or through secretion. But on top of those obvious changes in lipid metabolism, a tissue that looks like this must be metabolically different than a healthy tissue, not just for lipid metabolism. And we decided to look at early stages in MASLD during steatosis, simple steatosis, to take a deep dive and look into the metabolic changes that occur in the disease, because these would definitely then set the stage for disease progression and whatever events occur afterwards. So our model system for steatosis was a very well described one, the Western diet model in mice. We have a preclinical model. So you feed the mice a diet that's compared to the chow diet, which is rich in carbs and fiber. The Western diet is rich in fat, in fatty acids, and in sugar and sucrose, and also has cholesterol. The mice are very happy to eat it, and very quickly, after several weeks, 10 to 12 weeks, you can see very clear liver steatosis. There's an increase in body weight, an increase in liver weight. There's dyslipidemia is apparent from higher cholesterol, circulating cholesterol. Liver enzymes are increased, indicating that something is happening in the liver. And indeed, at the end point, we see, when we look at the gross anatomy of the liver, we see this pale color characteristic of a fatty liver. And histological analysis show the characteristic lipid droplets, and you can even be sure that these are lipid droplets with all red O staining. There are, at this stage, no signs of inflammation or cell damage, considerable cell damage. So we have simple steatosis. We then use this model to do untargeted, or I can say unbiased, metabolomic profiling of the liver, comparing the chow diet and Western diet, and also plasma between the two groups. The way we did the analysis looked only at polar compounds. So this excludes all the hydrophobic compounds, like the lipid, the triglyceride, the cholesterol, other sorts of fats, which would obviously would have been different. But when doing a polar analysis of polar compounds, we are including all the central carbon metabolism, like glycolysis and the Krebs cycle, amino acid metabolism, nucleotide metabolism, all the central metabolic pathways are included. And principal component analysis show that we are looking at two distinct metabolic profiles in both tissues that we analyzed between the Western diet and the chow diet fed mice. We were able to identify about 300 compounds in both tissues. And doing pathway enrichment analysis, metabolic pathway enrichment analysis, demonstrated that the top-ranking pathways were related to amino acid metabolism, mainly glycine and serine metabolism. So now we went ahead and did a targeted metabolomic analysis, meaning we specifically looked for glycine, and we could see that it was significantly decreased both in the liver and in the plasma of the steatotic mice. And if you look through the literature, you can find actually quite a lot of evidence. It's not sometimes pointed out, sometimes it's kind of a byproduct of a different analysis. But you can see that glycine is consistently reported to be decreased in various metabolic conditions, including mast cell, atherosclerosis, the metabolic syndrome. And this is reported both for preclinical models with different diets, even for different mouse studies, but also in patients. And as amino acids go, there are reports of other amino acids being altered in mast cells, but this is like branched chain amino acid, but they're reported to increase. And glycine is the only amino acid that is consistently reported to decrease in metabolic diseases. And from a biochemical point of view, it's not really clear why glycine is decreased. In our particular model, in the diets, the protein content was the same. So dietary glycine levels shouldn't have been different between the two groups. That couldn't be the explanation. And glycine is a non-essential amino acid. It should be synthesized quite efficiently in the body. So it was quite intriguing for us as metabolic people. What would be the mechanism to explain this consistent and widely reported effect? So if you think of glycine, its level could, or any other metabolite, the levels could decrease either if it's produced less or consumed more. And glycine is an amino acid, has a lot of pathways, metabolic pathways that can synthesize it in the body. That's why it's a non-essential amino acid. And I teach my students in the biochemistry course that glycolysis, for example, is not just a pathway to make ATP, but also a pathway that can make several amino acids, and serine and glycine are included. So, for example, one of the main pathways to synthesize glycine, as described in all the textbooks, is from serine. It's an offshoot of glycolysis. 3-phosphoglycerate, a few enzymatic reaction is converted into serine, and then serine, through an enzymatic reaction catalyzed by an enzyme called SHMT, is converted to glycine. There are other pathways to make glycine. And so that could be one reason why glycine is low. Or, as I said, it could be consumed more. And again, glycine is involved in many metabolic pathways that use it as a substrate. And actually, serine is also a substrate, is also a product of glycine, because the SHMT reaction is reversible. Glutathione, which I'll touch upon shortly, is also downstream to glycine and kind of really piqued our attention. So when we looked at downstream metabolites that glycine serves as a precursor for, we could see that all the ones that we could think of were decreased in the same way that glycine went down, its downstream products went down. So it couldn't tell us that this was the reason, because something is generated more, except for serine. Serine was the one that was different. Levels were not increased in the liver and were actually even elevated in the plasma. So we decided to take a closer look at the serine-glycine interconvention catalyzed by the enzyme SHMT. And for these, we gave the mice with the different diets labeled glycine. So it's labeled with a non-radioactive isotope of carbon, so carbon 13 instead of the all common carbon 12 that makes up 99% of the carbons in our biosphere. And this carbon 13 has a higher molecular weight, which could then be detected in the mass spectrometer. So we gave the mice labeled glycine, which means glycine has 2 carbons, as you can see here, and we chased to see where those carbons end up. So here, at the end point, we could definitely see that mice that received labeled glycine have higher levels of glycine, and most of this glycine is the labeled glycine. It has an M+2, mass +2, because the 2 carbons are labeled. But what was more interesting is to look at the serine. And we could see that the labeled carbons contributed to a considerable fraction of the serine in the Western diet fed mice, in the steatotic mice. This is just an hour. So very rapid de novo synthesis of serine. And another point to make, so glycine was used to make serine, but another point that I want to make, that again, if you consider the biochemistry, and I won't get into it, glycine has 2 carbons, serine has 3 carbons, so 2 carbons of glycine plus 1 carbon makes serine. That 3rd carbon can also come from glycine through a different enzymatic pathway called the glycine cleavage system in the mitochondria. And then we can get serine with all 3 carbons labeled, or serine with just 1 carbon labeled. So actually, this would mean that this labeling pattern of serine would mean that 2 units of glycine are consumed in order to make 1 unit of serine. So not only is more serine synthesized, but once you do the math and calculate how many glycine molecules were used to make the serine, the difference between the steatotic mice and the healthy mice is even more pronounced. So that would be a good indication why glycine is decreased, because it's used to make more serine. And in order to see if that was the case, we treated the Western diet fed mice with a specific inhibitor that inhibits SHMT. So the glycine to serine route is now blocked. And we gave them the labeled glycine. So this is only Western diet fed mice. So the ones that did not get the inhibitor have a lot of labeled serine in the liver, and no de novo synthesis of serine was present in the inhibitor. So this glycine, as expected, contributed to serine synthesis, also same in the plasma. What's really was nice to see that once we blocked this pathway, glycine levels increased. So we came to the conclusion that it's the increase in serine synthesis that causes the glycine depletion we see in fatty liver. So it's not that serine is used to make glycine in the way we teach in the biochemistry book, but in the liver, it's the reverse SHMT activity, and this consumes glycine, decreasing circulating and liver glycine levels. I'm not showing it here, but we were also able to show that the serine is then used to make sphingolipids, which are synthesized de novo from serine and palmitate. That's the first step in sphingolipid metabolism. And fatty liver is characterized by elevated sphingolipid metabolism. So we think that's where all the serine is going, or considerable fraction of the serum is going. I want to ask a different question now. Okay, so we see that glycine is depleted. What is the consequences of that? What would be the downstream effect of the low glycine that is so widely reported in other mice models and in patients? And I told you that a major substrate that's synthesized from glycine is glutathione. So glutathione is a tripeptide. It's made up of 3 amino acids, cysteine, glutamate, and glycine to make up glutathione. And glutathione is a major antioxidant in the cell, in any cell, but also very important in the liver, and oxidative stress we know is an important aspect driving fatty liver disease. So the glutathione has a reduced form, a monomer, and then it can reduce reactive oxygen species, ROS, and become oxidized, GSSG, a dimer. When we looked at glutathione levels in the liver, and indeed in the steatotic mice, we could see that it is significantly decreased. And of note, the other 2 amino acids that are used to synthesize glutathione were not altered. It is only the glycine that was lower. So we think that glycine decrease limits, or at least in part limits, glutathione de novo synthesis. So the obvious question for us, what would be the consequences of this low glutathione in the steatotic hepatocytes? So the liver is a major hub, I don't need to tell you, a major hub of drug metabolism, of xenobiotic metabolism. Glutathione plays a very important role in these reactions. And the paracetamol metabolism is a very classic example for this. So paracetamol also doesn't need any introduction. I'm sure we've all came across it and probably used it. It's a over-the-counter painkiller and antipyretic, so used to treat fever, used all over the world, safe in indicated doses, but its overdose, paracetamol overdose, is a major cause of acute liver injury, of drug-induced liver injury, and also, therefore, a major cause of acute liver failure and an indication for liver transplantation. Just for terminology, we in Europe call it paracetamol. In the exact same compound, same drug, is called acetaminophen, and then therefore it has a shorthand acronym called APAP, which I will use for the next few slides. So how is paracetamol metabolized in the liver? Most of it through phase 2 reactions, if you're familiar with the term, glucocoronidation mostly, and also sulfonation. And these conjugates generate a non-toxic compound that can then be excreted very efficiently in the urine, so no problem. A small proportion is metabolized through a phase 1 reaction, generating a problem. NAPQI is a very reactive free radical that can cause damage to the cells, but luckily hepatocytes have glutathione, and it reacts spontaneously or enzymatically with NAPQI and forming an adduct that is then again non-toxic and excreted in the urine. The problem starts in cases of an overdose, when because of the high doses of paracetamol, more NAPQI is formed. this leads to any glutathione depletion. There's just not enough glutathione to neutralize it. And then there's this very reactive free radical going around in the cells, causing oxidative damage, which could become very severe and could lead to tissue failure several hours later, and the next day after the actual molecular events have taken place. So if someone comes in to the hospital and tells them that they think they've taken a paracetamol overdose, an APAP overdose, sometimes it's intentional. Lots of cases are intentional. Some cases are unintentional because this paracetamol is found in a lot of other drugs or taken chronically. The standard of care is to support synthesis of more glutathione, and that is done by giving them N-acetylcysteine. And as I said, cysteine is one of the amino acids used to synthesize glutathione, and that leads to an increase in glutathione to deal with the NAPQI, and it has to be done in the early hours after the overdose has been taken, not when the damage has already been done by the ROS. So we said, okay, with a tissue with less glutathione, like MASLD or steatotic liver, would be more sensitive to paracetamol. And quite surprisingly, I think, because MASLD is very common and paracetamol administration is very common, despite that, there aren't a lot of real clinical studies to check if there's increased sensitivity in NAFLD, in this case, to acetaminophen overdose, to APAP overdose. Most of the literature kind of quotes this one study, which is really big, lots of tests. This was done about 20 years ago in the United States. They looked at reported cases. It's a retrospective study, so they looked at reported cases from 1997 to 2005 of acetamine overdose, hospital admissions, and they looked at the registry codes to see which patients were also diagnosed with NAFLD, as it was called then. But this registry code, which I think is maybe used by the insurance companies there, covers everything under the NAFLD umbrella except cirrhosis. So this could also be late stages of steatohepatitis. And I expect that there were not, because of the underdiagnosis of early stage simple steatosis, I expect most of the patients were not early stage if they got the diagnosis, because of course no additional diagnosis was done at the time of the treatment for APAP. So with all these limitations, they do see that there is an increased sensitivity, increased hepatotoxicity, or increased acute liver damage in the NAFLD group. Another reason, another kind of red flag why I think most of these were cases with quite advanced NAFLD is that even though it was a huge study, they pulled tens of thousands of medical records. Only 0.1% of that huge population of the study was diagnosed with NAFLD. And even the authors comment that the incidence is much lower than expected from the demographics included in the study. So there is definitely indication that NAFLD leads to increased hepatotoxicity of acetaminophen, but I think it doesn't really answer what happens at early stages of the disease. So we wanted to take a closer look at the link between glycine, glutathione, and APOP toxicity. We first looked at using cell line model, an in vitro model. It's a hepatocyte cell line. It's a mouse cell line, but it's non-transformed, unlike HepG2, for example, which is transformed. So it's not cancerous. It's immortalized, obviously. But we like these cells because you can load them with lipids. You can incubate them with fatty acids, and they're very happy to take them up. And then you can see the intracellular triglyceride content increasing, and the palmitate content increasing in these cells. But the reason we really like them is that they kind of recapitulate the metabolic phenotype that we saw in the tissue. Just the incubation with palmitate leads to a decrease in glycine and also a decrease in glutathione. So we said, okay, so they also have low glutathione. How would they respond to acetaminophen? So we looked at the oxidative stress in these cells following treatment either with acetaminophen alone, palmitic acid alone, or both compounds. And we could see this is a stain, D-hydroxyethidium, a stain for ROS, for superoxide, that the APOP-induced oxidative stress, the palmitate-induced oxidative stress, and we had a kind of a combined effect of both agents, which also manifested in the cell viability. So you can see that cells that have palmitate are more sensitive to APOP than cells that do not have palmitate loading. What about the mice? So we took our chow diet and Western diet fed mice. We gave them an overdose of APOP, not an extreme overdose. It's twice the maximum single dose recommended in humans. So for humans, the maximum single dose is 1 gram and a daily dose of 4 grams, so twice that as equivalent in mice. So both groups had elevated liver enzymes, right? There was some toxicity even in the chow diet fed group, but it's clear that the steatotic mice had a much more extreme effect when looking at circulating liver enzymes. When you look at the gross morphology, we can see hemorrhaging. That's why the pale color characteristic of the steatotic liver now becomes as if it's normal, but it's because of increased hemorrhaging, and histological analysis also demonstrated increased hemorrhaging in the steatotic mice. So it's definitely more sensitive to the same dose of acetaminophen. If we look now at the glutathione, we see that they start out with a lower glutathione, as we know in the Western diet mice. 3 hours after, both groups have almost negligible levels of glutathione, but at the recovery, we can see that the healthy mice have consistently higher levels of glutathione, and their glutathione recovery rate is faster. And they indeed, if you look at the 6-hour time point, you can see that the redox stress in the Western diet fed mice is higher. And this was also evident by activation of the transcription factor NRF2. We could see downstream genes of NRF2 being more activated in the Western diet fed mice. So yes, they are more sensitive to the drug. Is glycine really the underlying cause? If we treat them with glycine, will this go away? So Going back to the cell model, now this was actually another project, so this is not lipid-loaded cells, but we could show for another project that in cells that have low glycine, if we give them glycine in the medium, they take it up, and this supports de novo synthesis of glutathione. This was done now with labeled glutamine. Okay, so M+5 labeled carbons. We could see that glycine supports glutathione synthesis in the cells model. And in the mice, this was done only on the Western diet fed mice. So we gave them the same dose of APAP as before, but also glycine immediately afterwards, and also at the 3-hour time point where glutathione levels were extremely low. So mice that got glycine obviously showed higher glycine in the liver, but what was important and encouraging was that they had higher levels of glutathione. And when the glycine was labeled, we could see here that the glycine labeled carbon ended up in glutathione. So this administered glycine supported de novo synthesis of glutathione. We could see a decrease in the redox stress markers, the GSSG to GSH ratio, and this is another metabolite that's indicative of oxidative stress, also decreased. What about the clinical parameters? The ALT, AST levels went down, and liver hemorrhaging was also improved. And the mice survival rate the next day was the same as the chow diet. So yeah, a picture is worth 1,000 words. So this is the chow diet mice. We gave them a dose that they could handle, and they can manage very well with this overdose the next day. That's how they look the next day. But the Western diet fed mice with the same dosage look very poorly, very unhappy 24 hours afterwards. Steatotic mice, same mice, but ones that received glycine can now handle the same dose of acetaminophen much better and show higher levels of glutathione. So glycine administration did attenuate the APAP toxicity in the mice with simple steatosis. So to conclude, just a summary of the findings: mice with liver steatosis showed decreased glycine, and that was because the glycine was used to synthesize serine. The consequence of that is a low glutathione, and it impedes de novo synthesis of glutathione when it's required for xenobiotics neutralization. And we could see that elevated hepatic glycine, I didn't show you the knockout mice results, supports de novo glutathione synthesis and protects steatotic liver from acetaminophen toxicity. So basically, there's a balance between glutathione levels and tissue damage, and we can increase glutathione levels in the mice by giving them the glycine that was depleted. And it can suggest that monitoring circulating glycine levels should be considered in mast cell patients. The most important point that I want to make is that simple steatosis is therefore not clinically benign, even if it doesn't affect steady state liver function. When the liver comes under stress, the changes in metabolism that occur in hepatocytes can have very serious implications to the way xenobiotics are metabolized. So we need to give respect to this stage and consider it when we manage patients, not we. when doctors manage patients. And most of these results were already published in a paper in Cell Metabolism. And this work, I want to thank the people who did this work. Most of this was done by my PhD student when I was a scientist in the lab of Eyal Gottlieb in the Technion. So most of the work was done by Dr. Alia Gaev, who's an MD and now a PhD as well. And this was done with very close collaboration and discussions with our friends in University of Michigan, Professor Eugene Chen and their lab, and Professor Oren Rome's group in Louisiana State University. I've now moved and opened my own lab in Ariel University, and looking for curious students who are interested in studying liver metabolism and how that could affect drug toxicity and how that can inform us on developing a new therapeutics for this condition. So if anyone is interested, they're welcome to contact me directly. And I want to thank you, obviously, for listening, and I'll be happy to take any questions.